My name is Alexander Hurbic. I'm a PhD in AI and the CEO of Blackthorn AI. Today, I'm going to talk about AI and biotechnology application and real study, which involved two computational methods and one empirical wet lab for confirmation for theoretical studies using mathematical models. Major problem: how AI and mathematics models correspond with the reality. Gartner has reported that 85% of AI projects won't deliver. That's terrible, don't you think? Here is example of a couple of links. You can find this information. So it's a not a fake. It was a widely spread around the world. You may conclude that, oh, I didn't have to believe this hype. I want to avoid this. AI even might be scared. Let us be constructive and check the sources. The real publication is very different from the thing that was spread around. Instead of 85% of AI won't deliver, the true news were that on February 13, in 2018, almost 4 years ago, Gartner statisticians predicted that though 2022, 85 of AI projects will deliver, not because AI is bad, but due to bias in data, algorithms, or the teams responsible for managing them. If you dig a bit further, you will find out that out of 100 failures in AI, the management is responsible for 77%. So don't believe the hype. Instead, don't believe anyone. Check your sources. Don't trust everything you hear and read. Ligand is usually a small molecule, which you see on the picture to the right. It changes the conformation of a larger molecule, which is a receptor. Change in a conformation results in a change in the function of the receptor, and usually it ends with a cellular response of some kind. A receptor is a large biomolecule. Typically, it's a protein or DNA. Ki is the inhibition constant. Inhibition constant is the measure of inhibition of a biological function of the receptor as a result of interaction with the ligand. Ki is the numerical measure of such change. Usually, ligand inhibits the receptor, means its chemical interaction potency goes down. The stronger the potency goes down, the stronger the function is changed. Usually, not always. We will use that later as a target for machine learning models. Molecular mechanics is an oversimplification of a chemical reality. Atoms are represented as elastic balls. Chemical bonds, which connects atoms in a molecule, they are represented as springs. Describe this system of atoms and bonds with a number of equations. We add them up, and this is how we compute the overall potential energy of the system. When the state of the molecule will be the most stable, right? When its energy is the lowest possible. Molecular mechanics serves the purpose of finding the optimal conformation for a molecule. If we minimize this function, radius, distance of bonds, and the locations of atoms as a parameters, you will be able to tweak them so that the energy is minimized. This is how we can simulate and compute the optimal structure of any molecule. Molecular dynamics is a computer simulation of the physical movements of atoms and molecules. It represented all the molecules as balls and springs and described the intermolecular forces like van der Waals and electrostatic. I'm letting the system to evolve over the time. As I observe how it behaves, I can just understand is it stable or not, molecules interact between them, and how and why and when. It was one of the methods we used in this study. You see trajectories on the image to the right in red. We are moving from state to state of the system, changing coordinates of atoms, trying to numerically solve Newton's equation of motion. Let's get back to the current study. There were some receptors and some ligands which were interacting, and we try to assess those interactions using multiple methods. The receptor was glucocorticoid receptor binding domain, or GR. We studied how ligands interact with this receptor. It has been observed by a group of scientists that these small peptides inhibit the cancer cells growth. We had to understand to which receptor they bind to and the mechanism of interaction. Small peptides as ligands, and we compared their strength of binding with the glucocorticoid steroids. They were used as a control. It means if these ligands bind to this glucocorticoid ligand binding domain or GR, their strength of interaction is sufficient to confirm the anti-cancer biological effect. Let's consider methods. The first method is the receptor ligand graph neural network. We call it RLGNN. So first of all, it takes as input atomic coordinates of a receptor, and then it takes a SMILE string responsible for the ligand. Those small peptides and glucocorticoids were represented as SMILE strings. Then we convert a receptor atomic coordinates to a connected graph, which encodes the secondary structure of this protein. Edges and the nodes are all numbered. This is how we know what amino acid is connected to what amino acid and in what order. Also, we encode the protein receptor on an amino acid level. We do not go deep to the atomic level. We interpret a receptor as a set of amino acids which are binded between them, so that we describe the bond, the order of a connection, and we encode the amino acid with a set of parameters. All of that encoded as a graph. As for SMILES, we represent SMILES later as a structure, and we also encode that as a graph. But comparing to the receptor graph, the ligand graph is made of atoms as nodes and atomic bonds as nodes. And we parameterize each atom with such features as a molecular mass, valency, number of electrons on external layer, and others. And we parameterize bonds with such parameters as a type of bonds. It's a singular, double, triple bond. Is it aromatic bond, and length, and also hybridization type. So it means that we create mathematical models of a receptor as a set of amino acids and a ligand as a set of atoms and bonds. We hope to model the reality in that way because real structures, real coordinates, real compounds are very similar to these graphs. So our intention was to mimic the reality as much as possible. Now we have two kinds of inputs, encoded receptor and encoded ligand. And we pass them as an inputs to arms of a huge graph neural network, transform them to vectors, and then we concatenate the receptors and pass them to a multi-layer perceptron and trying to map to a logarithm of inhibition constant. The logic of a graph neural network is hidden behind and oversimplified here. As for data set, we had 5,300 protein ligand complexes from the protein data bank, along with the corresponding inhibition constants. Similar protein ligand complexes with recorded inhibition constants and train null model to learn the dependencies. In silico screening method number 2 was molecular dynamics. Molecular dynamics currently is a gold standard for such in silico studies. It is reliable enough. It predicts affinity and interactions in a 99 or more cases, if you know how to use it, of course. Our sequence was the following. First, we took this GR receptor and put it to Autodoc VINA to find a proper initial position of a ligand. Autodoc VINA is good at prediction binding site, the position of a ligand, but it is not used to compute the energy. It is good for initial position prediction. Let's talk about image to the right. Here you can see the spatial 3D structure of multi-sites structure and something inside. This structure is the volume where we model interaction. And inside the target receptor, which is GR in our case, and the water floats around small red things. Actually, it's a water. The water is also modeled in molecular dynamics because typically in natural biological systems, everything interacts in a solution, and it's a solution of water with some vitamins, ferments, and other things, but the base of it is water. Dots of red and blue are ligands. So we take this frame, we fill it with water, we pull our ligand and the concurrent ligands, our biological molecule, which is a GR, and then we shake it for some limited amount of time, which is measured in nanoseconds, because all the things move very fast and it's hard to model on the interactions there. We set the temperature, we set the pressure, set the kind of a thermostat. We chose the method for molecular dynamics to be GROMACS 2020. And the force field, GROMACS 54H7 force field. And the third one was